Bioinformatics. Without any delay, I would like to start a session with positivity. So now I call upon Muthu Lakshmi of second year Biotechnology for prayer song. Now I would like to call Halima of third year Biotechnology to give a welcome address.
Good evening to one and all gathered here. On behalf of our college management, our beloved chairman, Sri Raja Rao sir, our principal, Dr. Ramesh Babu sir, our director ma'am, Dr. Biola Devamalar, head of the department, Dr. Arshana ma'am, our beloved teachers and fellow students. I, Muhalima from third year Biotech department, extend a warm welcome to all of you present here on this webinar, Bioinformatics Research and Applications by Dr. Noble Kurian, Assistant Professor at Department of Life Science in B.S. Abdur Rahman University of Science and Technology. I also want to extend a note of thank you to the speaker and the principal of Prathyusha Engineering College for accepting the invitation. I feel privileged to have introduced you all to the honorable speaker. The Department of Biotechnology was started in 2001 and is offering B.Tech Biotechnology program since 2002 and its M.Tech program since 2019. Our department has signed MOUs with various companies and organizations like Slick Alpha Private Limited, BG Crops Private Limited, RNA Bioscience, Synchromax, Abex Biotech, LifeTech Research, Medox Biotech, etc. The department has produced 100% results in academics consistently and produced 6 university rank holders of 16 to 20 batch in 2019 to 2020. The department has also produced 100% placement records of students in various companies like Monash Biotech, Zippo R&D Solutions, Capellan Sterile, Pulses Health Tech, and Episodes Private Limited. The students of the department has received Best Paper, Best Project awards by participating in various international and national conferences. Our department has received various research grants since 2019 to 2020. The department has active EDC club in which our students have won TNSI Student Innovator Award with a cash prize of 1 lakh. The department has Bioinfotech club in which currently 85 students of all years are actively involved in club activities. They also have published paper, carried out mini projects and published it in international journals. And at the end of the session, we make memories and grab knowledge from the guest lecture with great enthusiasm. Before we carry on with this event, I would like to express an appreciation to all the people who made this occasion a reality and this institution successful. This would have not been possible with the sheer motivation of you all. Without any further delay, I would like to start with the event. We would be delighted to have your suggestions and questions at the end of the event. Thank you.
Thank you, Halima. Now here we have Kirti Meenakshi of final year Biotechnology to give an intro about our guest of honor.
Good evening, one and all. Our resource person, Dr. Noble K. Kurian, is currently working as an assistant professor in BS Abdur Rahman University of Science and Technology, Chennai. He has completed his BSc and MSc in Biotechnology in University of Kerala and his PhD in melanins from bacteria in Cochin University of Science and Technology. He has also completed his postgraduate diploma in bioinformatics in Center for Bioinformatics and Computational Biotechnology. He has various research experience India has also worked in National Institute for Interdisciplinary Science and Technology from 2009 to 2010 and has carried out research in isolating characterizing bacteria from deep sea sediments in 2012 to 2016 and in Cochin University of Science and Technology. And he is also involved in culturing and isolation of membrane vesicles from Staphylococcus in 2017 to 2018 in National Institute for Research in Reproductive Health. He has worked as an assistant professor in various colleges such as SNGAC Arts and Science College, RNB Global University, and Atmia University. He has also worked as a guest faculty in Mahatma Gandhi University and as a faculty instructor in the Institute of Biotechnological Research. He has received awards such as Best Graphical Abstract Award and Best Poster Award, and also got CSIR Net Lectureship. He has done 23 publications in peer-reviewed journals with 1,462 citations with H-index of 10 and cumulative factor of 25. He has written various book chapters and also released a book named A Complete Lab Manual for Biotechnology. He has also presented keynote talks and has also participated in various poster and oral presentation in conferences. We are very honored to have such a knowledgeable person with us today, sir. Thank you.
Thank you.
Thank you, Geeti Meenakshi. Without any delay, we will start the event. The session is over to you, sir.
Yes, thank you, Halima, Yamini, and Meenakshi for giving a nice introduction. So, thank you all people who have invited me. It's a great honor to present in this Bioinfotech club. I have gone through your Facebook pages. All I have seen very big personalities present through this club. So I also has given an opportunity. Thank you all of the biotech department, especially HOD and all faculties in the department, as well as the students for inviting me, especially Praveena madam has invited me through LinkedIn, and thank you ma'am also. So I am not spending much time in that, and I will share my screen now. So here internet problem is there. If any interruptions come, please inform me in between.
Okay, sir.
So hope you are seeing this presentation.
Yes, sir.
Yes. So let's study about bioinformatics. I can't see the who all are present or who all are asking questions. I will answer the questions last. So I'm just presenting. Okay. I will answer the questions last because I can't see what is happening in there. I'm just presenting. Okay. So everyone are bioinformatics students or biotechnology? So everyone here are bioinformatics or biotechnology students? So
We have a
anyway,
subject for bioinformatics, sir.
bioinformatics students are there. Okay. So you people will be familiar with most of the tools and techniques in bioinformatics, and I am trying to make it into a how it can be utilized in research. Many things will be familiar to you. I am not much in going much into tools or techniques or anything. In a research oriented, application oriented, how what is bioinformatics, that is what I am going to share with you some things. So basically bioinformatics means you will be studying in textbooks that it is a combination of computer and mathematics and information technology and biology, all will it's a combination which forms the bioinformatics. For me it is usually as computers after 1980s the computers have revolutionized the whole world. Everywhere the computers came. So generally biology, in biology also there is lot of data. In order to store that, analyze that, retrieve that, we use computers. It is not just for biology. Chemistry also use chemoinformatics are there in the chemistry. Even commerce used computers. So every field, as computers came, every field used utilize the advantage of using computers. So biology, when coming to biology, we have a lot of type of data. as the sequencing technology got emerged, as after the human genome sequencing has done, okay, during 2003 the draft genome was published. After that there was after that the technology of sequencing has been greatly what evolved. So during that time the bioinformatics, the field of bioinformatics got evolved. So let us go through a brief history. I think you people will be familiar with this picture. We will just go through a brief history. Brief history is the bioinformatics. I am just with this history I am going to say the what is bioinformatics. So this history will tell everything about bioinformatics. So bioinformatics, the field is developed around 50 years, maximum 50 years. I think after computer came, that computer technology flourished after 85 only. This technology has been highly flourished. So we can say 50 years is this technology has, or bioinformatics has its age, we can say 1970s to 2020s, this during these times. So in 1970s itself, the before 1970s itself, the term bioinformatics was coined by scientists. Actually, they used the term bioinformatics to just to just indicates the biological information which is collected through different experiments. It is not about the bioinformatics which we see today. Now the bioinformatics which we see today is what just revolving around the central dogma. You people all know central dogma. What is central dogma? DNA to RNA, RNA to protein. So most of the bioinformatics revolves around this central dogma concept. So at that time, the term, when they are coining the term bioinformatics, it's just meant that information from biological experiments, all this. So just they coined the term bioinformatics early, just before the 1970s itself. But actually the bioinformatics is the field was evolved, and it now it is mostly revolves around the central dogma. We study about DNA sequences, RNA sequences, expressions, as well as proteins. All these we have studied by using bioinformatics. So we can see, firstly we can see, I think you people will be studying about the sequence alignment algorithms, such as Needleman Smith Waterman algorithms. These algorithms is the ones first evolved in bioinformatics. So they have found out the algorithms in order to compare we can't say sequences. Its first genome was sequenced during the first genome was sequenced in mid 70s only. So they have found out this algorithm in order to compare data, biological data. So later we can see the so I can't what I can't explain everything, every application. So during this I am trying to explain most of the things, covering most of the techniques and tools in the bioinformatics. So please listen to that. Maybe I will take around 1 hour, 1.5 hours. It's okay. 1 hour, 1.5 hours maybe I will take mostly. I'll try to how much what is the duration of this talk? Can you tell anyone? Actually, before starting I need to, hello, anyone there?
Yes, sir.
I am what is the duration of this talk actually? Can anyone tell? Okay, I will take 1 hour, more than 1 hour. Okay, so I will be continuing. Okay. So protein data bank was formed in 1970s. The protein data bank, as you know, protein data bank mainly consists of the protein structures, protein, DNA, RNA structures. Mainly the structures were derived from experimental methods. Which experimental methods you use for protein structure determination? mainly X-ray crystallography, NMR spectroscopy. These are the two methods which we use for determining the protein structure. If it is the protein is crystallizable, we use X-ray crystallography. If the protein is not much crystallizable, usually it is not much crystallizable, we use NMR spectroscopy. Nowadays, more advanced techniques has came, such as cryo-electron microscopy can be used for determining protein structure. So these all structures are deposited in this protein data bank. It's a primary database which stores the protein information. This was one of the database which evolved first. Database, what is database? Actually, database is database actually collect data. Okay, databases usually collect data, organize data in proper way, just like library. So they organize data, and when we need the data, we can retrieve. We usually use the term download the data in the case of what other websites, movies, all you will download. So just like downloading, we use the word retrieve the data. So we retrieve, we store, we store, and the storage is organized in such a way that it can be retrieved, it can be downloaded, it can be retrieved easily. So that is the database. So one of the structural database is the protein data bank. It is formed during 1970s. You can see Sanger method is one of the first sequencing method. It is considered as the first generation sequencing method. This Sanger method is a dideoxy sequencing method. I think you people are familiar with, because I can't explain everything. I will just go through an overview. So Sanger's sequencing is a dideoxy sequencing method, which is which was considered as first generation sequencing method, which was evolved with the Maxam and Gilbert sequencing method. This both method was, Maxam and Gilbert sequencing method is done by chemical modification. These both methods were considered the first generation sequencing method, and it takes time also for sequencing. So we have sequenced smaller genomes first, and then the PAM matrices got evolved, and then Smith-Waterman algorithm got evolved. It's a local alignment algorithm. The PAM matrices are going to used to score the alignments. So all these got evolved nearby 80s. So nearby 80s only we know that I think we people have not seen much computers during 80s. So mostly this all development occurred in US and that places, so where the computer has been highly evolved. So 80s and nearby 80s, the first algorithm as well as the blast algorithm, blast algorithm was produced developed during 1990s. So this was one of the, these two are one of the most widely used tools which is used for finding out similar sequences or finding for pairwise alignment. We use these tools. So these so this FASTA and BLAST algorithm was developed, as well as by using multiple sequence alignment we have constructed molecular phylogenetic tree during that time. Molecular phylogenetic trees, which tell about the evolution relationship between organisms. We use DNA sequences to find out the evolution relationship. So in order to find out evolution relationship, we as the sequences, more sequences came, we can see the sequencing of phage lambda occurred, and concept of sequence motifs, and creation of NCBA. That's so I will come to that. The phylogenetic tree, by the sequence comparison or sequence alignment, such as multiple sequence alignment, pairwise alignment, etc. People started constructing molecular phylogenetic tree using the neighbor joining methods. Different methods are there for constructing phylogenetic tree. One of the method is neighbor joining method. By neighbor joining method, people start constructing the phylogenetic tree. So then came the NCBA. I think you all have bioinformatics, biotechnology. All people are familiar with NCBA. It's a website which is formed by NIH, is maintained by National Institute of Health USA and and National Library of Medicine NLM. This website contains a lot of databases which contain sequences of nucleotide, protein. I'm not going through anything. I'm just going through the overview. I think everyone is familiar with this NCBA, all these things. So nucleotides, proteins, sequences, gene sequences, genome sequences, literature data are present, and books are present. Then tools such as BLAST, what SNP databases, and then what a lot of databases are present in this. A lot of databases, tools such as primer BLAST is present in order to find out the suitable primer, and there are what ORF finders are present, and then BLAST is present. Different types of BLAST is present. So then the BLAST algorithm was developed, and BLAST became the most common tool. This Altschul et al. Has developed this BLAST. This is one of the most widely used bioinformatics tool, and it's one of the most cited tool also. And for surprise that this tool was first published in a journal which is around 4 impact factor or something like that. Now it is 4 impact factor. It is published in a small journal, not like not any Nature or any big journal. So this BLAST algorithm was published, and then BLAST is one of the commonly used tool since now. Also it is developed during 1990s, and many genomes such as Saccharomyces cerevisiae, Haemophilus influenza genomes got sequenced, and then came the human genome project from 1990s. As the sequencing technology developed in around 1975, that time people were thinking about sequencing the whole genome. We were sequencing only small genomes here, phage genome, yeast genome, all these things only. But we were thinking about sequencing the whole genome of humans. It was a very big task. We thought about sequencing in 1990s in US government with different countries, collaboration with different countries, Japan, China, and different countries, France, and they have done this human genome project from 1990 to it had taken 13 years to 2003. 13 years with Celera Genomics, a company also was involved. Craig Vender's company Celera Genomics also involved in this project, and they have sequenced the whole genome by the old sequencing technologies available at that period, and then came the next generation sequencing technology that had changed the whole bioinformatics scenario. This has created a lot of job opportunities for bioinformaticians. So as you people, I think you will be familiar with next generation sequence data analysis and its opportunities. You already know, I think. I hope you know it. So next generation sequencing technology start with this instrument from Roche, the 454, which work with the principle of pyro sequencing. Usually during the addition of nucleotide, their pyrophosphate will be released. Usually this pyrophosphate, what is principle behind this Roche's 454 sequencing is that this pyrophosphate will react with an enzyme called luciferase, which will create color, a fluorescence, which is used to detect which base is added. So actually I think you people will be familiar with the next generation sequencing. It will be conducted in flow cell or wells, etc. It will be a massive parallel sequencing, a small slide in which there will be lot of wells in which the sequencing of a lot of millions of DNA fragments is carried out parallelly at once. So in each well, one nucleotide is added at a time, and so that from which well the fluorescence fluorescence come. Based on that, the sequencing has been done. So the sequencing based on pyrosequencing, the release of pyrophosphate from the pyrophosphate from during the addition of each nucleotide. This pyrophosphate usually it will react with luciferase enzyme and create a fluorescence, which is used principle behind. Later principle behind the 454 sequencing. Later came the several sequencing method in 2000, nearby 2010, 6 to 10. Solid is one of the sequencing method where no DNA polymerase is used. They use a ligase enzyme from sequencing. And then came the ion torrent. Ion torrent, there is no fluorescent dye was used. Instead of fluorescent dye, they used pH change. During the addition of, as I said, during the addition of any nucleotide, pyrophosphate is released. Less like that, during the addition of nucleotide, H plus ions is also released. As H plus ions are released in the solution, we know that H plus ions will determine the pH of that media. So the pH, the change in the pH is determined using a sensor, which was the principle behind ion torrent. So basically this is the next generation sequencing, and many genomes have been sequenced, and human genome, because of the next generation sequencing, the sequencing speed also increases, and more genomes have been. Nowadays genome sequencing is nothing, because every day thousands of genomes are being sequenced and deposited in genome databases. So now it was nothing too big to be said. So actually many Drosophila genome sequence and human genomes, this genome sequence published during 2003 was it is not a complete human genome. Later many versions, many assemblies came. You can see, check the genome database such as UCSC genome browser or any genome database and see. I think now it is 2013 assembly is the active one. I think new assemblies are there, but we use 2013. I think I need to recheck. So there are different assemblies still updating regarding human genome, and later the new sequencing technologies came, which are PacBio and Oxford Nanopore. Nanopore, you can see it is a small sequencing machine. It's very small, with less than a mobile. We can keep it in our pocket and go. So just like a mobile, we can use this Oxford Nanopore sequencing, and PacBio sequencing has also revolutionized the DNA sequencing. It makes the sequencing fast, and it's not just fast. Actually, I have not go to rice genome also sequencing in between. So actually, when we came the first generation sequencing to the next generation sequencing, such as 454 solid ion torrent, actually the problem was we can only sequence a small segment of DNA, about 150 base pair, like that. The read length, we usually use the term read length. The read length was so small, so that a lot of read will create, and it will assembly of the read all will be more tedious task. So the read length was increased by the PacBio as well as these Oxford Nanopore sequencer. They try to increase the read length, thereby making the sequencing more efficient, and this read length was increased in the case of PacBio, and in Nanopore they have not used even a what they use a protein nanopore. Inside there will be electric current, and when the DNA is passed through that, based on DNA is passed through that nanopore, the change in electric current occur. The change in electric current for A, T, G, C are different. Based on that, the sensor will sense and find out the sequence. So all sequencing technology is changing. We are finding out sequence of bigger reads. I am dreaming that one day we can sequence the whole genome from the start to the end with a single machine, without being assembly or anything. There will be something we are going to develop. Just a vague thinking. The bioinformatician, you people will be crazy about that. I am not much a bioinformatician. I am teaching bioinformatics for a long time. That's all. So like that. So as the COVID-19 came, a lot of the sequencing technology has developed, and a lot of job opportunities are also there for bioinformaticians, because a lot of changes were happening in this variants, sequence changes were happening in the variants of corona. So for analyzing all, lot of opportunities are there for bioinformaticians also. So I hope you got a basic idea of what is bioinformatics. So that's the history, that is the bioinformatics, which I hope I have covered most of the bioinformatics. So let us go to Dr. John's laboratory. It is not my laboratory, this Dr. John is a hypothetical man. So I have just taken a picture from internet. So I think he looks smiling, so he has his own laboratory, and John was uh, what is happening in John's laboratory is running a multidisciplinary laboratory. So it is a hypothetical person. So just we can go to his lab and see what he's doing. Okay. So John, please follow me. If before starting that, I should check whether you are online. So because I need to. Okay. So in between, I don't know whether the internet is connectivity is proper or not. So that's why I'm. So if I am explaining and you are not hearing, there's no use in that. So John, in during February 2000, at that time John was sitting in his lab, and he was saying that his corona is very much problematic. Corona infection start, and it is increasing. Coronavirus is everywhere. Hmm? Coronavirus is creating a lot of problem, and everywhere mask is being introduced, and corona is creating a lot of problem throughout the world. Everywhere it's becoming a pandemic. So actually, Dr. John got a interest in this corona research, and he thought that we need to design some drug to inhibit the coronavirus. So he just studied the earlier viruses which are similar to that of the corona. Earlier SARS virus, MERS virus, all are there. So he studied, and he just and so from other researchers suggestions, and he just found out a protein called spike protein, and researchers has already found that spike protein is involved in binding with the AC receptor of human lung epithelia, and then thereby descending to the lung epithelia, and killing the lung cells. So this lead to the this is massive destruction of lung epithelia. So there was a suggestion from the scientists that if we can block this spike protein, it can by using some drugs, it will be helpful for preventing this corona. So this was one of the hypotheses which many scientists put forward. So mainly the problem was any problem there? Are you following?
Yes, sir.
Okay.
Yes, sir.
The problem was we don't have actually we need the structure of the spike protein to find out the drug. You people all know that docking, all these things, you people will be very much familiar. So I don't want to explain much on that. Anyway, we need the structure of the spike protein. So where we get the structure of the spike protein? Corona is very much increasing. So getting the structure, we need experimental methods, such as X-ray crystallography, NMR spectroscopy. We need to crystallize the protein, and then finding the structure. It will take a lot of time, but people are dying. He can't. John can't wait for that. And then came the sequencing technology, the NGS, all technologies were I developed nowadays. So he can very easily sequence the whole phage genome, sorry, not viral genome, coronavirus genome, and he got the genome, and he got the DNA sequence. Yes. Any problem? Some sound is coming. Okay. So he got the DNA sequence. Then he was thinking that from DNA sequence, how we can find out the structure. Then there are you people know that from the DNA sequence we can easily find the protein sequence. There are a lot of tools such as XPC tool. Lot of tools are available which can convert the DNA sequence to protein sequence. So he found out the spike DNA sequence. From the spike DNA sequence he can find out the protein sequence, and from the protein sequence he found out that he can design, or he can model the structure of the spike protein. He can model the structure of the spike protein using in silico tools or bioinformatics tools. So you don't need to wait till the structure is available from the experimental method. So he can model the structure. Luckily he got a spike protein, similar spike protein from the earlier SARS or MERS, and so that he can use homology modeling for the modeling. Earlier SARS or MERS spike is available, the structure is available. Earlier we have determined the structure of earlier SARS or MERS using X-ray crystallography or NMR spectroscopy, etc. So, and their structure as well as a sequence of protein is available based on these template. These using this structure of the earlier SARS virus spike protein, using this as template, he tried to design, try to model the spike protein of COVID-2, the corona virus, COVID-19 virus. So he used the template earlier available similar protein template for designing the or for modeling the structure of his protein, of which structure is not known or structure is not found out in by any experimental methods. So he got the structure. Luckily he got the structure. Now he can find out he can try different drugs using in silico techniques such as molecular docking, etc. He need to analyze the drugs by their ADME properties, all these things. After that he can check the drug by the modeling, the molecular docking, etc. Actually, if the SARS virus, the spike protein was not available, this SARS virus or MERS virus spike protein was not available, or any similar protein is not available, what he will do? He will go for a de novo modeling. It's using different algorithms. We can model it from the scratch. De novo modeling he will go for. Luckily he got a model which is similar. If any model, experimentally determined model which is similar to that of the experimental related model is available, and the sequence of this model will be, if it is similar to that of the sequence of the sequence of our unknown structure unknown protein, we can model it using the homology modeling. If we don't know any similar structures, we will go for de novo modeling. So then anyway Dr. John got this structure, and after the structure he studied different drugs. He just shortlisted different drugs. We can find a lot of drug molecules in KEGG is one of the websites, is one of the pathway database. We can find a lot of drugs you can download from the KEGG, as well as you can download the mainly we used to download many drug molecules from the PubChem database. I think all you are familiar with all these words. PubChem database you can download, and we can check their drug properties. And otherwise, if you don't have any, if you are trying to try a new drug, you can draw in ChemDraw or any other software tools, and you can if you have a drug, you can determine its structure, and we can use that structure for you can use that structure to find out how good it can bind to the spike protein, and it can prevent the action of the spike protein, or it can prevent the function of the spike protein, so that we can get rid of the corona. So then comes the molecular docking for Dr. John's help. He has shortlisted many drug candidates, and he had tried to find out how these drug candidates binds to the protein, or is spike protein, is protein of interest, how strongly the protein binds, where it is binds, and he can shortlist a lot of what a lot of places in which this drug can bind, and he can finally come to a conclusion where it can bind properly, or whether it can bind properly and stably, so that it can be used as a proper drug. So this molecular docking help in the for this molecular docking helped Dr. John. I think you are familiar with all these databases. I think I will go through this. So you will be familiar with the Expressy Translate tool. I will just go through and just show and go, because I am taking, because I am just presenting is not good. So Expressy Translate tool, if you just enter the DNA or RNA sequence, you will get the protein sequence. You can just paste it like a blast or like that, and you will get the sequence. And then what we have discussed here, then we can see the Explicit Translate tool, and then homology modeling. Different tools are available. Simpler tool we can see, Swiss model is one of the simpler tool. We can very easily model Swiss model. It's one of the simpler tool which can easily we can use to model the structure. You can just paste your target sequence or alignment, target template alignment, the sequence of the target and template can be aligned and can be paste there, or you can add the target sequence so that it can be sequenced. Sorry, it can this if you just paste your target sequence and search for the template, the tool itself search for the similar proteins, similar templates. If it is more than 50%, 40-50%, you can go for the modeling with that 40-50% similarity is there within their sequence and the template they are choosing. You can go for building the model. You can get a model and you can analyze the model further. So these are the things which and then different docking tools are also available for docking. So this company has a Swiss dock, another tool is there. You can easily dock online, but it's not it is not much advanced like Schrodinger or Autodock or any advanced docking tool, but you can easily dock, easily upload your you can just submit docking, click submit docking, so that this is very easy tool. You may have played with this. Yeah, I'm just, as I am presenting there, I am just showing. You can target, you can select, and before selecting the target, you need to do a target preparation. You need to remove the ligands, het atoms, and you need to remove water molecules from the. You can use any softwares. I am using Discovery Studio for that. You can use any softwares which you are using, and you need to go for a ligand preparation also. After that, you can upload without preparation with the ligand or water molecule. If you upload the structure here, you will not accept that. After preparation of the sample and ligand, you can just go for docking, so that they will find out. It's very easy tool. It's not may not be advanced like other tools which we are using daily. So you can use this tool for docking purpose. You can use this. So like this, Dr. John has done all these experiments in his laboratory. So this is all these things. I will just, so he have found out the structure, he have found out the protein from the DNA. All these things he have done. Okay, so docking he has done, homology modeling he has done. So all these things he have done using these tools which came to help him. So Dr. John was a multidisciplinary, his lab is a multidisciplinary laboratory. He is not just studying viruses, he is just studying bacteria also. He was working on a bacterial amylase gene. So he just isolated amylase gene, means using a PCR he has just amplified amylase gene, and then he was thinking that he got only some ATGC, he don't know whether that gene is amylase or not. He just designed the primer and just just done the PCR and sequence that. He don't know, yes, he got some ATGC, he don't know what is amylase or not. So actually in this situation what he need to he think is that if there is some similar sequence to that of the amylase is available, I can compare that amylase, my amylase gene, with the similar amylase gene from other organism. He is working on bacillus, let us think, and if we have pseudomonas, we have some other vibrio amylase is available. He is thinking that, Dr. John is thinking that if this E. coli, pseudomonas, vibrio amylase is available, and so that he can compare his sequence with that amylase from other bacteria sequence, then he can find out whether is the ATGC series is amylase or not. So he found out in rescue, the most familiar tool is that the blast. He found out, you all know that, but so he found out that whole amylase gene from even humans, even bacteria, even every organism is found in is collected in a particular database called GenBank, which is maintained by NCBA. He came to know that, but he is thinking that NCBA, the GenBank contain a lot of amylase gene or a lot of gene other than amylase, many genes. How he is going to find out everything? Yes, Google in web world, a lot of things are there. How we can find out information regarding your college, your department, Prathyusha Engineering College, and with your department? How we can find out? A lot of colleges data, lot of things are available. So there you need a search engine, that is Google. You can just type in Google, so that you can easily find out about your department, about your teachers. Every information will be directly directly sent to you. Just like that, this NCBA, this GenBank contain a lot of information. In order to find out the gene similar to that of the amylase gene, which Dr. John was isolated, what he need to do? He will paste this amylase gene sequence, which he got in the blast search bar. Like you paste in the Google, you will paste this blast search bar and just search it, so that this blast will search throughout the whole billions of sequence data and find out whether his gene is amylase or not. The blast usually do pairwise alignment. Blast usually compare between two sequences. The process is called pairwise alignment, and find out how much similar it is. It will align the sequences two at a time, so that it will find out how much similar they are. So I'm just going with a similar term. There is a lot of differences between similarity, homology. So a lot of things are there. I'm just using the terminology similar between the two sequences. So So then after finding out his amylase gene is similar, his gene is similar to that amylase from different bacteria, amylase from humans. Then he had a thinking that whether we can take human amylase and bacterial amylase, fungal amylase, and all different organisms, C. elegans amylase, and whether we can compare this all these amylase together. So he went for a multiple sequence alignment MSA, where you use a tool called Clustal Omega. So this Clustal Omega, which compare more than 3 sequences, and it usually it's algorithm compare 2 sequences at a time. And what this algorithm do is that this algorithm first construct a gate tree with and it will try to align most similar sequence first and less similar sequence to the last. So like that this algorithm go. So this Clustal Omega, earlier it was called Clustal W. So this Clustal Omega will compare more than 3 sequences, but this algorithm has limitations. It can only compare about 20 sequences because more than 20 sequences is not possible in the case of our computers can't compare. Our computers, there is a limit in what these comparisons. There is a limit in this algorithm. It can only compare up to 20 sequence. So 20 sequence can be compared using the cluster omega. And while comparing the sequence, it can be connected to molecular phylogenetic analysis. Usually cluster omega, as they do the multiple sequence alignment, they itself give a phylogenetic tree. The phylogenetic analysis can also be done by comparing the more than 20 sequence. We can find out how much they are evolution related, thereby using different techniques such as neighbor joining, maximum parsimony, maximum likelihood, UPGM, different methods we can construct. We can select the methods, they all have some limitations, advantages. Depending on the methods, we can construct some are character based, some are some make use of the evolutionary relationship. So we can construct this type of phylogenetic tree which will tell about the evolutionary relationship, and there will be a root in the phylogenetic tree and which will ends in nodes and branches. We can see these organisms are more related, as we can see Bacillus series or Thuringiensis all are more related. And you can see these Bacillus species series, these strains are more similar. Like that, all similar sequence come with in a common node, from a common node. So like that, these are the phylogenetic tree. This can be constructed. I usually use a mega software for constructing a phylogenetic tree. Depending on you people, you can take a good phylogenetic tree from the Clustal Omega also. So all of these tools, I think you are familiar with this, and I will just go through and just we can see that, and just I will just go through as I am just going through. So we are very much familiar with BLAST. It will be available in NCBA website only. I will not make you boring, because BLAST, without knowing BLAST, no biotech student will be so there without what is I can't get, my internet is not going. BLAST, some slow, it is slow, it is showing. So I think you will be familiar with BLAST, Clustal Omega, as well as different phylogenetic tree constructing tools such as Mega. You can use sorry, my what is happening? Yes, stock is coming, but I can't take. Okay, I think now it is okay, I think. Okay, I will okay, I will go to the presentation. It's some problem with my presentation, my web browser. Okay, sorry, I can't. I think you are familiar with all these tools. So we can see, otherwise I will show you all these tools as we goes on. So blast and cluster omega, so by just taking the working on the particular gene, Dr. John had find out their similarity with other sequences, and how much it is similar to the other sequences, and he constructed a file. He compared with the how much they are evolutionary changed. You can see different genes, such as even hexokinase is a gene which is involved in the conversion of glucose to glucose 6 phosphate. It is present in every bacteria to human. So there is a lot of evolutionary changes have been happened from the hexokinase sequence when it came to humans. So you can find out a lot of changes have been occurred in the, these all enzymes function same now also, same in humans as well as bacteria, but a lot of evolutionary changes have been happened in these sequences. By using these multiple sequence alignment, we can find out their evolution relationship and how much changes, the mutations or changes have been occurred in these sequences, and we can construct the phylogenetic tree also with that. Okay, I'll just go through here and just I will check again, because I have prepared in such a way that I can show you blast, but nothing big to show you blast. I knew everyone know, but some problem with my if I close my meeting will be gone, so I am going with the presentation, sorry for that. So Dr. John was again involved in different multidisciplinary research, and he had a sequences. He had a sequenced genes in hand, but he don't know whether the where the gene start and ends, uh, where is the initiation codon, where is the termination codon of the gene. And when he tried to find out the genes, exons and introns in prokaryotic genes, then also he find difficulty in finding. It is only ATGC, we can't find out which is in introns and exons. We know that this in the splicing junctions we could know that there are specific sequences are there. Using that we can predict the genes, which is exon, which is introns. We can predict ORFs. These introns, exons can be predicted by tools such as GenScan, all these. You can just simply give the sequence, you will get the result, but I can't do that now. You can get it. The tool will find out the exons, introns, and if you can just enter your sequence in NCBI or a finder or some tools like that, we can get the ORFs. And usually ORF is the open reading frame. What is open reading frame? Usually open reading frame of a gene. Gene, we have a 1000 base pair, okay, a gene. For just you think, in 1000 base pair, a gene will not start from the first base pair and ends in the 1000 base pair. Gene usually start maybe from the 400 base pair and it ends at the 900 base pair, okay. The rest of the sequences were regulatory regions, all these things, regulatory regions, promoters, all these things. So if it is 1000 base pair, maybe the gene will start from 400 base pair and end in 900 base pair. So in order to find out where the gene start and where the gene ends, in order to find out the initiation codon, termination codon, all this, ORF finder is a very good tool we can use. It will search for the 6' translations and find out which of the ORF is the right ORF. Usually bioinformaticians go for 6' translations. Means if there is a I hope you all know it. So if there is a sequence called A, T, G, C, G, T, A. So how we will try to translate is that he will start from this frame, okay, 2 frame. First frame is this, and you can just see, just fast I am just telling because I think 1 hour is over. So we will just the second frame we will start from here, this, this, this. Actually third frame we will uh, start from the third nucleotide where it is here. Third frame we will start first, second, third frame we will start from here, GCG, TA what it is, TAG it is like that. So we will start from here. So 3 frames we will get it, 3 positive frames, plus 1, plus 2, plus 3 frames we will get here. Then we will go for the complementary sequence of this. Okay, we will just write down the complementary sequence as it's taken new smaller sequence just for writing this only. T A C G C A T C. Okay, complement, and then we will take the reverse complement of this. Okay, I will write reverse from here. Okay, I am going to write C T A CTA C G C T A. So we will take the reverse complement. Then we will start the same thing. First frame is we will start like this. Second frame what we will do? We will start from the second nucleotide. Second frame we will start from the second nucleotide. I will just show, because just lecturing the things is not good, that's why you all get all these things from the internet, so there is no use in telling the applications just in a plain way. So so we will start from that, these things, this second nucleotide will start the second frame. The third frame will start from here, this way, this way, this way. So like this, this frame in the reverse complement is denoted as -1, -2, -3. In the this strand the frame is called +1, +2, +3. So 6 frames translations bioinformaticians will consider, and from the 6 frames actually nothing is that 6 frames is that usually in the we don't know, there are DNA have 2 strands. Okay, actually the 6 frames is that DNA have 2 strands, we don't know which strand, from which strand the mRNA is produced. Okay, the gene can be produced from either of the strand. So that is the kind of principle behind the 6 frame translation. So we don't know which gene is produced, this gene is produced from this strand or this strand. So for that only we are just creating the 6 frame translation, and from the 6 frame translation only one translation will be the correct translation which codes for the particular gene. So the ORF prediction can be done by ORF finder. You can just check the ORF finder, just enter your sequence just like a BLAST or any other tool, just enter your sequence and just get the result. You can see these translations +1, +2, +3, -1, -2, -3, all these things. So gene prediction, I will just winding up. Primer design, maybe I think 1 hour is over. I can take time.
Yes, sir. Yes.
Okay, okay. So I am just making it more more time is taking. So okay, primer designing we can use when you are doing molecular biology experiments, we can design primers using bioinformatics tools. There are lot of tools such as primer blast is there, primer 3 is there, lot of tools. You can just enter your gene sequence. Usually what is usually when we are going for amplification of a gene, okay, gene is here. Okay, what we will do is that double stranded DNA, okay, gene is here. So what we will do is that we need to have the primer in order to get the full gene. If you are just want to identify the gene, you can just select the primer inside the gene, but if you need to get the full gene for cloning, all these purposes, the primer should be outside the gene, otherwise the gene may not be functional. So a primer should be designed in this flagging region. This is outside region, this is a gene. So in the outside region we need to decide the primer. So we should download the sequence. You can download the sequence from UCSC genome browser, etc., by just with the flanking region. We can download the genes from the UCSC genome browser with the flanking region, so that we can just download the sequence and just enter it in the primer 3 and give your parameters. Primer designing, there are a lot of parameters are there. Your primer should have this much more than 50% GC content. It should be around 20 base pair. More than 20 bases should not be there, and it should not have a high amount of repeats which can form hairpin, or it should not be base paired. This primer should not form primer dimers or anything. So lot of parameters need to be taken in mind, and we can design the primer. These all parameters will be taken in mind by the primer 3 or primer blast, etc. They can design the primer, and this primer, after designing, you can check it by in silico PCR. I think using genome browser as a in silico PCR, as well as NCBI also has a in silico PCR, I think so. So UCSC genome browser has a very good in silico PCR. You can just give your primer and try to check whether your gene has been amplified in silico. If your gene is amplifying in silico, you can give the primer to the people who synthesize the primer and use it in the wet lab and for the amplification of the particular gene. So primer designing is one of the Dr. John has used this technology for his research. So when he was he had some gene sequence. When we are try to find out when he has some gene sequence, and he need to clone that gene into some vector. So in order to find for that, he do not know, he has only some ATGC. Again I am saying ATGC only. He don't know where which enzyme he need to cut, which restriction enzymes are the enzyme used to cut the DNA, which is involved in the cloning of DNA into which help in cutting the vector as well as the DNA, which help in the cloning of the particular gene. So he actually he don't know what restriction site, what restriction enzyme he can use. So the bioinformatics again came helping him. So there is tools such as NEP cutter is there. New England Biolabs, that is a company which usually it's more than just half of the is advertisement of this company, because this company is manufacturing a lot of these restriction enzymes. So we can put our gene sequence in this just like BLAST or any other tool. You can put in this NEP cutter, so that you can find out whether your sequence contain any restriction site, which enzyme you can use it for use for cutting your gene. So he had done his cloning also. He is very much happy. I can see from his face. So when he is trying to study, because the research is not just doing things in the laboratory. It is we need to read a lot for getting new ideas, all these things. For reading, you can you people MSc students, you can read. You may not get most ebooks. You can rely on bookshelf from NCBI. Bookshelf is containing very good books, and it is published in HTML format. It is very easy to read than a ebook. Ebook we need to zoom it and read very pages. But in a bookshelf we can get the book in a very good layout, HTML layout, and it is very easy, very good to read, very good for our eyes also. And Dr. John was involved in reading the research papers using PubMed. PubMed is that literature database which is present in NCBI, where we can get a lot of research papers, and I think Google has also Google Scholar, all are there. We can go for literature databases. And in order to know about the diseases, all these things, genetics of diseases, there are databases such as Online Mental Inheritance in Man. OMIM is there. OMIM was first maintained by NCBA. Now it is a different site. You can go and check, and literature, a lot of literature about which can help you in different research in different diseases. When you are studying about different diseases, how it is associated with how the genetics of different diseases, and its pathogen, what its every symptoms, genetics, everything will be present in this OMIM. You can just check, it will be helping you in your research. And metabolic pathways, if you need to, any every compounds which we are studying, mainly biotechnologists, when you are studying the production of a particular chemical, production of a particular enzyme. So these all enzymes, proteins are all produced by different pathways. I am working on melanin pigment. Melanin, I think people have said in the introduction. So melanin is synthesized by a tyrosine mediated pathway. Tyrosine is a precursor. So different pathways are there involved in the production of all these biotechnological products. So the knowledge of metabolic pathways as well as the enzymes, proteins, all involved in this pathway is very important. So KEGG, Kyoto Encyclopedia for Genes and Genomes. So this KEGG website will give idea about the pathways, and KEGG nowadays KEGG has more data, more annotated data. Annotation is the word I have used. What is annotation? If there is any raw data, in order to find out different terminologies are there, I think you people will be very much hearing about the annotation. So annotation means usually I said ATGC only Dr. John have. From that ATGC we can derive a lot of secondary data, such as we can predict genes, we can design primers, so we can find their restriction site, we can find their promoter site, we can find their protein sequence. So from raw ATGC we can find out lot of other data, so we can find their motifs, domains, all these things. That all extracting data by in silico methods from the raw data. That extracted data is called annotated data. This process is called annotation. Actually curators do this process. Curators are the people who do this process. So NGS data analysis, so Dr. John is working in that also. NGS, next generation sequencing, as I said, next generation sequencing is done in a small chip, like just a glass slide which you use, that size only the chip has. Just checking whether everyone are there. What is this?
Yes, sir.
Everyone are there. Okay. I will continue. Okay. So next generation sequencing analysis, because I don't know my internet will cut or not, that's why I'm okay. Next generation sequencing data analysis, Dr. John was working on that also. He was he's in a headache that after next generation sequencing, sequencing, lot of data come, lot of DNA pieces, what to do, lot of DNA pieces around him, what to do, how to make it into a perfect final DNA form, what to do. So then also bioinformatics come to help him. The bioinformatics tools, such as different tools are there for making the usually what when NGS data NGS sequencing, what we do is that as I said, the read length is so small in most of the modern NGS data sequencing machine. So it can only read up to 150 base pair or 300 base pair, like that. Okay. So what we do is that we will break down our DNA into small sizes, so that it can be sequenced. These fragments DNA, our DNA is so big. Okay, 3 into 10 raised to 9 base pair. These DNA is broken down to lot of pieces, and all these pieces are sequenced parallelly in small chip. This will create a lot of data. Usually what we do is that before sequencing we will go for a library preparation. Library preparation is just we have a lot of sequence. After DNA fragmentation we have a lot of fragments of DNA. These fragments of DNA will be amplified first using a PCR. That's the library preparation. So after the PCR, and some adaptase will be linked to the fragments, so that it can be easily bound to the slide which we are going to do the sequencing reaction. This is all done in the library preparation. So lot of DNA is there. This DNA is being sequenced, which will create a very big data. That is why we say we are in bioinformatics, it's creating is very big data. These data need to be analyzed and made it into a human genome. It is very tedious task, and it is need a very good, very great computer. That much of big configuration computer. I don't know much about computer configuration. So that much of supercomputers are needed for analyzing this data. So there are a lot of tools which help scientists to analyze the data, such as star is there, BOTI is there, SAM tools is there. These are the tools which are these are the fragments. These used to make this fragment into the continuous DNA form which we can read. Okay, these are just fragments of DNA. We need to make it into the continuous form so that we can read, we can find out the sequence of a particular chromosome. This chromosome sequence, the chromosome will be broken down into number of pieces. So we need to assemble this sequence. For assembling this sequence, we use all these tools, and there is a web server called Galaxy is there. Usually these star, BOTI, SAN tools all work with the command line interface. So command line people, I think you people are familiar with you will be interested in all our bioinformatics people interested in command line. You are familiar with and you are interested in using command line tools, but many people like me who are not good in programming, so we need to go, we are just in that jail of Microsoft, in the GUI so that jail, so we can't come out of that GUI, so we don't know the command line interface. So we use a web server called a Galaxy, which just like NCBI, it contains a lot of number of tools which involved in the analyzing the data which is coming from the NGS machine. Actually this is the pipeline of NGS sequence data analysis from a Illumina machine. From Illumina machine, usually the sequence come in a format called BCL format, which will be converted to a FASTQ format. Usually FASTQ format is the format which we use in the sequencing, and I will show the FASTQ. So I think you are familiar with the FASTQ format, where I can show. Where is my PPT? Yes, this is some other workshop PPT. So this is the FASTQ format. You can see Illumina FASTQ format. You can see the sequence come like this. There will be a sequence identifier. 4 lines are there in the FASTQ format. I think you are familiar with the FASTA format, greater than symbol and the title, and then comes the sequence in the next line. Here the FASTQ format, it consists of 4 lines. First line is a sequence identifier. Next line next line is the sequence itself, and the third line is just what is happening. Third line is just a separator, and fourth line is gives us the quality of the sequence. So quality scores will be given by the in the FASTQ format. Quality of the sequence is being given in the FASTQ format. In FASTA format, there is no quality or anything, just sequence only. So FASTQ format consists of the title, the sequence identifier, which contain the data about the sequencer, which terminality is used, different things are there. So usually in Illumina, the sequence come in a BCL format, which will be converted to FASTQ format. This result come in a FASTQ format. Then comes the sequence, then will be a separator will be there, a plus sign will be there, followed by the sequence quality will be there. The sequence quality will be denoted in ASCII symbols. So the ASCII symbols you can get it from here. So depending on that, different scores have been given. The ASCII characters is represented. So all these things you all know it well. So basically the NGS sequencing, what we do is that people who don't know, I am saying, so in Illumina pipeline, what we do is that we will go after getting the sequence from the sequencer, we will go for a fast QC QC means quality control. We will just go for the checking the quality control, whether the sequences are good, whether the sequences are contain adaptive sequences. Usually we attach adaptive sequence to the DNA. So we will check whether the adaptive sequences are there. If any sequences are not good, overlapping peaks are there or not good, we will remove it by a tool called Trimomatic. So if any adaptive sequence or any other things are present, we will remove it using a tool called Trimomatic, and then we will go for the genome assembly. What is genome assembly? We have only the pieces of genes. These piece of DNA need to be assembled. How we can assemble? We can use different tools such as spades in the case of Galaxy, or different tools, star, bote, charm tools, all are used in the genome assembly. So the genome assembly, we do it in two ways. If we have a if Dr. John has sequenced a human genome, he can use this use the reference human genome which is sequenced in 2003 or 2013. That reference human genome he can download, and using the reference he can find out where the pieces are belong, where the DNA pieces are belong. Thereby he can build up the whole DNA. He can build up the whole DNA. Or else he is going to sequence a organism, there was a big elephant was there, what is his name, I forgot. So a fossil elephant, so I forgot that name. So if it is we are sequencing the genome of that elephant, we don't have a reference genome there. So then we need to look for overlapping sequence, and we need to assemble the sequence without the reference genome. So like that, if there is no reference genome available, we need to find out, we need to assemble the genome using overlapping sequence. After finding out the whole genome from the start to the end, then we will find out if there are any variants are present, SNPs, different variations are present in the genes. We can find out using variant calling tools such as SNP and various tools which we use. So different tools are there in command line interface as well as Galaxy. This is the pipeline of Galaxy only. So further the data will be visualized using different tools such as JBrowse or different tools which we use. So you can visualize the data. So these are about the DNA data analysis, NGS data analysis. So Dr. John is got the help from Bioinformatics in doing all these things. So important thing nowadays, not nowadays, it is very important for a bioinformatician. It is very important to study some of the programs. So Python, R, Perl, Java, these programs need to be, you will need to be thorough, for it need to be, it will help you in data analysis as well as developing new tools as well as software. So in order to excel in bioinformatics, you need to know these languages such as Python, R, Perl, Java. They all have this bio related BioPython is available, BioConductor is available, BioPerl, BioJava, all are available. So in order to deal with our sequences, all these things, and also Linux knowledge is also very important. Mostly the bioinformatics tool softwares work with a command line interface, because it is very, when you put a DNA for when you put a genome for analysis in Galaxy, it will take a lot of time. So when it's very easy, when you use command line interface, it is very easy compared to that of the GUI, and GUI tools are not available in bioinformatics. Most of the tools uses a command line interface. So for as a bioinformatician, so you should Dr. John is also improving his skills, and is studying, going through these all languages and trying to excel in bioprogramming. So then comes the AI era. AI is everywhere. what GPT and all this AI era is going to come. So bioinformatics also AI is going, or machine learning is going to come in bioinformatics, and it's not going. It's there itself. Many tools are being developed in order to predict different things. What about AI means? We have usually bioinformatics problems can be solved with the help of this artificial intelligence. Actually, we thought and we with our brain only we are developing algorithms, statistical tools, statistical things in order to develop various tools. Now we are teaching this computers, and by using that machine learning, the computers are making our what computers are mimicking the neural network. Artificial neural networks are mimicking our brain, and they are trying to make the processes that make the bioinformatics processes more simpler and faster than earlier. So now AI is going to come in protein structure prediction, homology modeling, and multiple alignments, phylogenetic construction, genome sequence analysis, all gene finding, all these areas. This AI is going to come. So you people will be eager to know about their career prospects. I am just saying about some the career prospect which you already know, but you can be a research scientist and research associate. So every biology lab nowadays now need a bioinformatician. It's very rare. Every biology lab, whichever they do, whether they do plant research, animal research, veterinary research, any type of lab, they need a scientist from bioinformatics. They need a lot of people who can handle their data. That much data is coming in each lab. So so you should be a multi-dimensional people, multi-dimensional, so that you can do wet lab as well as bioinformatical analysis, handling big data. It will help you in growing as a very big scientist. So you can be a professor like me and your teachers, so you can teach what knowledge you got. It is one of the what biggest profession, and I think you people also value the profession. So then you can be a data analyst. You are getting a lot of big data, and this data can be analyzed whichever the lab or laboratories. It can be a hospital, it can be, you can get a job in hospitals, you can get a job in these sequencing centers. Many places you can get opportunities as a data analyst. Lot of places, lot of opportunities you are getting as a data analyst. And you can be software developer if you are very good and we were crazy about the bioinformatics. As I have not said first, bioinformatics is a field where a core information technology, so software engineer or a mathematician can directly enter, and if he can or she can study some biology, he can, or he or she can be a scientist in bioinformatics. Bioinformatics is the area where a biologist can enter. If he or she is very interested in software, he or she can develop into a software engineer or a software developer. So if you are interested in mainly in softwares, you can go into a very you can develop these tools and help these big companies developing very good tools. And there are you people are also developing many tools. There is a it's a sea of tools are available in web, but developing the very quality tools are also matters. So you can focus on that. So thank you. This is, I think I have gone through most of the things, and most of the bioinformatics things, and their career prospects. Thank you everyone. Thank you for giving me an opportunity, and you can ask any doubts. If I know, I can discuss with you.
Thank you, sir. We had a great session today with you, and we have a takeaways in molecular docking basics and next generation sequencing and career prospects in bioinformatics. So let's have a questionnaire session in this, sir. So guys, if you have any questions, you can post it in your